Hi, my name is Xun Xun Shi, and I am a biomedical engineering student from the University of Ottawa, and I am here to present my work on a supervised learning approach to predict ion mobility mass spectrometry response for different lipid stereoisomers. Glucosoceramide and galactosoceramide, which I'll refer to in this presentation as GLICSER and GALSER, are stereoisomeric glycosphingolipids. Naturally occurring pairs of GLICSER and GALSER can range from different N-acyl chain lengths between 0 to 32 carbons and can also have varying degree of unsaturation. Although the pair are nearly structurally identical, with the only difference being the 4' hydroxyl group in the equatorial versus the axial position, they have distinct biological roles. Simultaneous lipidomic assessment of GLICSER and GALSER pair is crucial for biomarker discovery and pathomechanistic studies. However, simultaneous quantification of the lipid pair is difficult, as they have the same mass-to-charge ratio and retention time. One method to achieve this is by using differential ion mobility spectrometry, or DMS, which applies a set of voltage to provide orthogonal separation to ESI-MS-MS. With that being said, selecting the optimal DMS parameter value is challenging. There are 6 different DMS machine parameter values that contribute to the ion intensity detected by the DMS. Each of these relationships will also depend on the chain length and degree of unsaturation of the N-acyl chain for the lipid pair. Because only one DMS machine parameter value can be changed at one time, the method development process is very labor-intensive, time-consuming, and expensive, as it relies on the availability of the synthetic lipid standard. For example, to determine the optimal parameter values for separating galceramide D18160 and glyceramide D18160, each of the lipid species must be directly injected into DMS-MS-MS, and 2 experiments must be performed while the compensation voltage, or COV here, is ramped. The relationship between ion intensity and different COV value can be represented as an ionogram. Repeating this experiment for different separation voltage, which is a SV value here, allows for the rise of 2 different peaks as selective SV-COV combination, which indicates optimal separation. All of these experiments will then need to be repeated for the other 4 DMS settings. Therefore, there is motivation for developing an in silico optimization method to replace these manual experiments. In our approach, we formulated the ionograms as Gaussian features. For both GLICSER and GALSER, we took the peak COV value from each ionogram as a mean and estimated the standard deviation value using the full width half-max formula above. We found that the standard deviation value did not vary greatly between lipid species, so we used the average standard deviation value across all lipids at each given SV value. The mean COV value did vary for different lipids and SV value, so we used a machine learning model to learn this relationship. Specifically, we used a neural network with 4 dense layers of 10 neurons each. The structure was determined during hyperparameter tuning with cross-validation. Prior to this step, we split the data into a cross-validation and a test dataset. The cross-validation dataset contains 14 lipid species that were used to select the neural net size and other optimal hyperparameters. We further evaluated the neural network on the test dataset containing the 3 Hodel lipid species. During evaluation, we regenerated the normalized ionograms from the predicted mean values and the average standard deviation value. Using our peak and valley detection algorithm, we identified all peaks and valleys of the empirical ionogram and the normalized ionograms. We annotated these ionograms as either separating or not separating based on the criteria that the valley intensity must be less than 50% the height of the lowest peak, and further use these annotated labels to measure the separation results. This is a result of the predicted test set lipid species using the trained neural network. In these plots, both of the empirical and the predicted mean values of the lipid pair are plotted against the separation voltage. Circles are for the predictions and stars are for the empirical. The black is for instances of no separation and color for the instances of separation. You can see that we're not only able to predict the peak COV value very closely, but we're also able to correctly predict for the range of SV where separation occurs. We're also able to predict separation of these lipid species at each given SV with very high accuracy, specificity, sensitivity, and F1 value. So in summary, we have shown the first machine learning solution that can be used to predict for optimal DMS machine parameter values for separating lipid species without the dependency of synthetic standards. I would like to thank and acknowledge all of our partners for sponsoring our work, and thank you for listening.